And the title of Dr. Ryan's talk is Colon Cancer 2013, Why It's Hard to Develop Drugs. Thank you, Dave.
Thanks very much, Don. Thanks for having me. I feel a little bit like this is I'm Mariano Rivera coming in firing some fastballs at you. This is a pure clinical talk. But I'm going to give you, having listened to Ryan Corcoran and the morning's talks and then Cyril's recent talk, I'm going to try and give you a flavor of why it's been so hard in colon cancer and give you a sense of where we're going in colon cancer. It's not that we haven't had success. It's that we've had some success, which has now muddied up the waters and making it very difficult for us to move forward. And oftentimes, what I find is that the drug companies talk at cross purposes with the clinical folks and with the laboratory scientists. And even though we're all speaking English, I'm not sure we're understanding each other. And so I've been spending a lot of time trying to figure out how to communicate better. And I hope I convince you that, that in fact, we can start to look at some areas in colon cancer. Ooh, let me try and get this forward. So every clinical talk, I always like to start with this. There's only 3 reasons for giving chemotherapy to anybody, and that's to cure someone, to help them live longer, and to help them feel better. And if you're not doing one of those 3 things, then you really shouldn't be doing anything. And there's been a lot of clinical trials recently, particularly in the colon cancer space, that you kind of scratch your head at and say, they didn't even try and adhere to live longer, feel better, or cure more often. And so when you're designing these clinical trials, you really have to move in that direction. This is the hit parade for the type of cancers that people die from. These are the types of cancers that we see every day in clinic. And I was struck by, I'm going to come back to a comment Jay Bradner said, which is, you know, even though you can slow down a cancer for 14 days in mice, in people, you really want to keep people alive month after month after month, and 14 days really doesn't matter. So that's kind of where we're coming from and where we've been. I'm in an area of GI cancers, and we've been, we haven't had too many home runs, I would say, in colon and pancreas cancer. And if you combine colon and pancreas cancer together, you can see that they're the 3rd and 4th leading causes of death for both men and women. So it's a huge unmet need right now. And I can tell you that I spend half of my time, I probably pitch 10 projects to pharmaceutical companies and others, biotech industry, and maybe we get 1. And even though it's the 3rd and 4th leading causes of cancer death for both men and women, for every 10 projects I pitch, I barely get 1. And that's probably overstating the fact. Everybody is scared of those two areas. So colon cancer, it's not like pancreatic cancer in the sense that we haven't had some successes in colon cancer, but the successes in colon cancer have kind of muddied the water so much that it's made it incredibly hard space to get a registration trial going. Some people have been able to do it though, and they've been able to do it mostly outside of the United States. When I first started back in '95, I was Bruce's first class of fellowship. And we had 1 drug, 5-FU, excuse me, 96. We had 1 drug back then, and that was just 5-FU. And the average person lived for about 6 months if you did nothing. They lived for somewhere between 8 and 12 months if you gave them 5-FU. In fact, I saw Jeff back there, Clark. We used to give 5-FU every which way, standing on our head, upside down. We gave it by continuous infusion, bolus, every which way you know how to give it, just to try and get people to live longer. But that's basically what we had. And then all of a sudden we had irinotecan, oxaliplatin, bevacizumab, panitumumab, cetuximab, and now we have Regrafenib and aflibercept just in the last 2 months. So how do we make sense of all of it? Well, the thing to note, and one of the things that Jay said struck home, and that was it's about living longer and keeping people alive. And in fact, we've been able to do that for a significant subset. When I used to first sit down with people with metastatic colon cancer, during that first visit, it was always really hard because you'd sit there and the median survival was 6 months if they didn't do anything. It was only about half of 1 to 1% of people lived for 5 years with metastatic colon cancer. And that's people who, we didn't realize at the time back in the mid-90s that you could resect people who had isolated metastatic disease. But only roughly 1%. So of the 50,000 people in the United States, right, 500 people lived for a long time, for 5 years or more, because they had a naturally slow-growing biology or because they had these phenomenally sensitive tumors to 5-FU. And it does exist. But nowadays, we're actually seeing about a median survival approaching 2 years. And we think that our 5-year survival, and this is from Scott Kopetz, put this together in the JCO last year, we think it's about a 20% 5-year survival. So we've actually had some success. If you think about it, the tail on the curve is not that bad. So I can actually sit in front of people and say to them when I first meet them, you know what, you got a shot of being around 5 years from now, a 1 in 5 shot, which it's not great, but it's not as bad as it used to be. It's not 1 in 100. So now that's 10,000 Americans every year of the 50,000 can hope to live for 5 or more years by going on and off all of these drugs. And that's the key point. They're going on and off all of these drugs throughout the course of their cancer. So where did we all start? Well, this is the mother of colorectal cancer studies. It came out about 10 years ago, and it randomly assigned folks with metastatic colon cancer to FOLFOX, IROX, or IFL. IROX was irinotecan and oxaliplatin, and IFL was the old way we used to give irinotecan with 5-FU before we realized it was too dangerous to do it with the bolus regimen. It turns out that we started at a year, we got out to about 1.5 years just without the biologics if we gave FOLFOX and irinotecan to everybody who had metastatic colon cancer. Of course, we dabbled in which should we give better, which regimen is better if we use an infusional 5-FU regimen, FOLFOX, FOLFIRI. These are all very familiar controversies to folks who were in the clinic at the time. It didn't matter. It didn't matter at all. You just have to give all the active drugs, and people did pretty good. Or they did as well as you could expect, and it didn't really matter in terms of the sequence of these particular drugs. Of course, what we didn't realize at the time, we got caught up in a kind of big pharma argument, was that it was all about the treatment discontinuation. Everybody kind of burnt out a first-line chemotherapy in about 6 months, and that hasn't changed. It hasn't changed not because the drug stopped working. It changes because our ability to continue people on more than 6 months of chemotherapy goes out the window. Very few people can stay on chemotherapy continuously for more than 6 months, at least this chemotherapy. But the problem was, and the problem with drug development, and the reason why everything is become murky, is because we actually see these kinds of responses. I didn't see these when I first started. And in fact, we then appreciated that you could resect people with metastatic disease. And so now, everybody who comes into clinic, we're shooting for this. We're hoping for this. And we know that this can happen. And the funny part is we actually don't know. We don't know what the really good biomarkers are for 5-FU, irinotecan, and oxaliplatin. We have some old ones that we think about, DPD, thymidylate synthase, ERCC1, but they really don't predict as well as you would like. And so we want to give everybody the chance to have this kind of a response. So much so that now the big thing that's happening out in the community, and I can tell you it drives me a little bit insane, is people aren't resecting or debulking for cure anymore. They're debulking to help people live longer, because that sentiment has seeped into the community. So now surgeons are going after peritoneal disease and liver disease. Radiation oncologists are doing SBR to lung metastases, or interventional radiologists are doing RFA to liver metastases, because one is growing and the others aren't. And that is making our ability to do clinical trials in this space, in the space of colon cancer, exceedingly difficult. So what happened? So we had anti-VEGF therapy. And anti-VEGF therapy we all thought was very complicated. The funny part it is very complicated. We still don't really know for sure how it works, and it is a very large controversy. But now we have 3 drugs in the space: bevacizumab, Regrafenib, and aflibercept. How do we get there? So the first study was the Hurwitz IFL study, and it was IFL plus or minus bevacizumab in folks who had first metastatic colon cancer in the first line. They hadn't been treated yet with anything. And it was dramatic improvement, almost about a 5-month benefit in terms of median survival. We've never seen that since then. Now, Herb got a lot of grief for this subsequently, particularly from this study, which is the euphemistically called 666 study, which was a Roche study looking at FOLFOX or CAPOX plus or minus Avastin. And this, because we had all switched to 5-FU and oxaliplatin in an infusional regimen, people thought you'd actually get a better benefit. Turns out that the benefit was in the range of about 2 months in terms of overall survival. And all the other studies that have been done with VEGF inhibitors, as far as we can tell, the benefit is about 2 months in the first line of therapy. And then the big question became, well, how's it working? Is it helping chemotherapy work better, or is it just an environmental effect? And we still don't know that, but we do know this year that just staying on a VEGF inhibitor seems to be a very good thing. Now, the reason why it's so hard to develop drugs, particularly in the first line of colorectal cancer, is this simple fact. Drug development companies do not want to wait 2 years for overall survival. If your median overall survival is 2 years, they then look for a surrogate endpoint. And that typical surrogate endpoint is progression-free survival. Can you imagine doing a first-line colorectal cancer study where about 75% of, or 70 to 75% of people come off of first-line chemotherapy for metastatic colon cancer for reasons other than progressive disease? I mean, how do you design the statistics for that? You're almost at the necessary numbers to do survival statistics. And so it becomes an incredibly difficult space to do metastatic first-line studies. And the reason they come off is because people just can't stay on the chemotherapy for more than 6 months. In the case of oxaliplatin, it's due to neuropathy. In the case of irinotecan, it's just due to sheer exhaustion of going through the treatment week after week. So what we learned this year, and we finally had some positive studies after years of negative studies, we learned that having some anti-VEGF therapy included all the time makes a difference. The type of anti-VEGF therapy probably doesn't make a difference, and that the original Hurwitz study was probably right, that if you keep VEGF therapy on board all the time, your overall survival benefit is probably in the range of 4 to 6 months on average over and above just giving chemotherapy. So how did we learn that? Well, first we learned the BEV beyond progression study. This was a study where patients in second line, after progressing on either FOLFOX bevacizumab or FOLFIRI bevacizumab, switched and were randomly assigned to either get bevacizumab or not in second line chemotherapy. And in fact, there was a survival advantage by about 2 months. So that's the BEV beyond progression. So continuing bevacizumab makes a difference. Well, at the same exact time, the aflibercept, which is VEGF trap, a protein that traps circulating VEGF, they randomized folks to FOLFIRI plus placebo or FOLFIRI plus aflibercept in metastatic colon cancer that had progressed on first-line chemotherapy. Now, what's interesting here is that not everybody on this particular study had prior bevacizumab. Well, how could they do such a thing, you ask? Well, they did it in Europe, where not everybody gets prior bevacizumab. And so the big question here is whether if everybody got prior bevacizumab would have had the same effect. And the answer is probably, based upon the BEV beyond progression study I just showed you, and 2, in a subset analysis of this study, having prior bevacizumab didn't seem to make a big difference. And so now you have 2 options of what to do in second-line chemotherapy. You can either let bevacizumab ride, or you can switch them over to aflibercept. I can tell you that they're having a hard time selling that to the community because the community oncologists get used to one thing and they just want to let it ride and continue because it's easy. So it's a really hard sell, which is making it really hard in the drug development world for us in convincing companies to go into second line, because now we have everybody going from FOLFOX Bev or FOLFIRI Bev and switching right over to the opposite plus bevacizumab. And so we're not even getting those patients from the community into the second-line clinical trial setting in the big academic medical centers, unless they start here with us. And what about Regrafenib? Well, Regrafenib, as you know, is just like sorafenib, and it is a dirty VEGF tyrosine kinase inhibitor. It hits a couple of other pathways, which I show up here. And what these guys did is a randomized placebo-controlled study in the refractory setting. So everybody had first-line and second-line chemotherapy, and it was powered for overall survival. And this is getting a lot of controversy in the lay press right now because it showed only about a 6-week survival benefit. But nevertheless, having a little bit of VEGF on board seems to be important, and it doesn't seem to be a at least it doesn't seem to be a cytotoxic drug, or even maybe cytostatic as much as it is environmental. There were almost no responses. In fact, placebo had a response, 2 responses actually, as well. So there you go. That's it for VEGF therapy. And the last thing is, well, why don't they try and displace bevacizumab in first line? Well, why would a drug company do that? Because it has become the graveyard for VEGF tyrosine kinase inhibitors. And the latest was just published last month in JCO, and this is FOLFOX Bev versus FOLFOX cediranib, which is the old AZ, I think 2171 compound. And you can see here that it was just not inferior to bevacizumab. So this has exhausted the VEGF pathway in metastatic colon cancer. But you can see that this rush into this space has used up first line and second line and third line patients. And it's very difficult to figure out what space you can get in as a registration study. So just to recap, VEGF works, and it probably does affect the general environment. The big thing with VEGF and where our research is currently is trying to figure out who is actually responding and what's the biomarker for response. There's been some data to suggest it might be circulating VEGF-A, but Janet Murphy at our center has been looking at this and looking at SNP analysis, and so far we haven't been able to find any specific group of folks that is having a great response to bevacizumab or any of the VEGF inhibitors. Throw into the mix the EGFR antibodies. Well, the EGFR antibody story is actually a little bit quicker. We gave it to everybody initially, and there was a very small improvement in overall survival. We quickly figured out that if you were KRAS mutant, you didn't respond. If you KRAS wild type, well, you might respond. Maybe you'll respond. But if you were KRAS mutant, you didn't respond. And that became obvious after the fact. We didn't, in the clinic, we're not as smart as you guys in the lab, so we didn't particularly pick up on that fact about the downstream activation. But herein lies the problem, and I'm going to come back to this notion in a second. You can see here in blue, these are the people who respond to cetuximab, roughly 15% of the population with metastatic colon cancer. Well, the population with metastatic colon cancer, as we're quickly figuring out, is roughly 10 different diseases. It's being defined by us in the clinic one way. It's being defined by the scientists another way. And there's a little bit of crosstalk right now, and it gets very confusing. So who responds? Those folks who are KRAS and BRAF wild type. The PIK3CA wild types respond better, as well as the folks who have PTEN. Increasing copy number of EGFR, those folks respond, as well as amphiregulin and epiregulin overexpressing folks. And certain EGF and IGF SNPs will respond better than others. The mechanisms of resistance are really poorly understood. We have a biopsy program going on right now to rebiopsy folks who've developed, were responsive and then acquired resistance. The purported mechanisms out there in the community right now are MET amplification or activation, and then perhaps driving some other mutations, activating mutations downstream of cetuximab or panitumumab. So right now, this is the kind of patients that we see in second opinion. It's maddeningly mushy. So a patient comes into us, and the fellow says to me something like this, or I go in and I talk to the patient, and they say something like this. And I try and piece it together. I feel like an infectious disease doc who's been called in to figure out which drug causes the low platelets. So FOLFOX Avastin, and then all of a sudden they can't stand the oxalate anymore, and they get CAPE Avastin, and then all of a sudden the cancer starts getting worse, so the oxaliplatin's added back, and then all of a sudden the cancer starts getting worse after a couple more months, and then they're switched to irinotecan Avastin, which is allowed to ride the whole way. And then all of a sudden the community doc says, you know what, I forgot to get KRAS status, let me go figure that out. They send off just to KRAS, they don't ask for anything else, because they're in the community. They get KRAS wild type, and then they're started on irinotecan and Erbitux, switch to panitumumab, which has absolutely no effect after people have progressed on Erbitux. We did that study. The response rate was 0. And then they're switched to Regrafenib, and then they come in to see us for, quote, phase 1s. So you can imagine being in biotech or drug, big pharma, how difficult it is to enact a clinical trial in the United States. So why is it so hard? Well, one, the line of therapy is incredibly messy. What's first line, what's second line, what's third line? But that's not really the drug development fault, and it's not really the community oncologist's fault. It's the FDA's fault for having such weird, rigid registration strategies or registration rules. The refractory setting is obviously the one to go into, but the refractory setting at that particular point, your median survival, once somebody has progressed through Regrafenib, you're talking someone's median survival is in the range of 4 months. How important is VEGF therapy? Can we determine if it's not important, can we just get rid of it and start testing some of these new products in a particular group of patients? Well, we haven't been able to, that's one of the reasons why finding out that biomarker status is so important for VEGF, who responds to VEGF, is so that we can move on with the other patients and figure out some new targeted pathways. It's difficult to evaluate in first line, nearly impossible for the reasons that I stated. And the other big issue is it's multiple different diseases. So when I first started, we thought it was one disease, and it was all based on the Vogelgram model. And what's interesting is that when you talk to the colon cancer gurus in the science field, in the lab, they're generally putting it into 4 separate categories. So the chromosomal unstable, the microsatellite unstable, the CIMP tumors, and then finally the IBD or chronic inflammatory tumors. What's interesting is that the colitis-associated cancers seem to be very poorly understood. They acquire multiple genetic alterations, and there's also a group of patients that have chronic subclinical colitis that go on to get cancer, and we actually don't know who those folks are because they're subclinical. The other folks have either a traditional pathway, a serrated pathway, or an alternate pathway. What's really hard, and this is where we start talking across purposes, is that they can have varying degrees of KRAS, PIK3CA, NRAS, and BRAF mutation status. They can also be CIMP high or CIMP low, and they can have different microsatellite status. Just to kind of drive home that point, Shuji Ogino across town looked at BRAF mutant as it related to CIMP marker status, and you can see that while CIMP low tumors were more likely, CIMP low tumors were more likely not to have BRAF. It wasn't exclusive, and it wasn't exclusive that all the CIMP high tumors had BRAF mutant disease. The scary part about this, and I'm going to digress here for 1 second, is when I was in medical school, they taught us that if everybody got a colonoscopy, we'd eliminate colon cancer. This was 20 years ago. So it came out the last 3 years that colonoscopies are not protective against right-sided colon cancers. Well, right-sided colon cancers, it turns out, so that the hazard ratio is literally 1 for a right-sided colon cancer and a prior colonoscopy. And the reason is that these tumors probably arise in polyps, or if you can call them polyps, they're flat. The endoscopist just can't see them. And these are most likely the serrated adenoma pathway, which then gives rise to usually CIMP high and usually BRAF mutation, but not always. We don't even understand the 2 major types of colon cancer, which are right and left-sided, let alone everything else. And so the biologic differentiation, when you start looking at all the mutation status and all the variations, I stopped here at 8 just because I was running into the MGH Cancer Center logo, but you could go on and on if you know what I mean. The other talking at cross purposes is when you're talking in the drug development world and in the clinic, you're talking about mutation status, which is very different. So we're then talking about folks who have KRAS, BRAF, PIK3CA, NRAS, and we're targeting those particular areas. But we think that they're these monolithic cancers, or these, homogeneous cancers. All the BRAF tumors are the same. They're absolutely not. In fact, I was just reviewing a paper, and this paper was looking at BRAF mutant colon cancers, and it was standard chemotherapy against BRAF mutant colon cancers. And the interesting thing is that I said, what was the microsatellite status on these folks on my review? And I got back, I wanted the immunohistochemistry. I want to know how many of these folks had methylation of MLH1. I got back a standard microsatellite analysis that they had done, and I didn't have the heart to ask them to go back and say, no, I meant CIMP and MLH1 and all that other stuff in the immunohistochemistry for the microsatellite proteins. So anyway, I told them to accept it. But this is my proposed design. This is the way we should be thinking about it. On the right is the traditional design, where you did a phase 2 study, and just for the sake of numbers, those of us who are MDs failed math, so everything begins at 100, because it's easier to do the percentages that way. In the traditional design, you enrolled 100 patients on a clinical trial. But the problem was you had 48% had wild type, 15% had PIK3CA, 5% had BRAF, 2% had NRAS, and 30% were KRAS. And so you didn't really evaluate the BRAF, the NRAS, and the PIK3CA patients. You got this overwhelmingly amount of folks who were wild type. In the new design, what we should be doing is enrolling these folks at least in the genotypes that we know, or at least in the protein pathways that we know. And I would argue that we should be getting these biopsies to post or in the middle of treatment to see which pathways we're actually turning on and turning off, so we can make some progress and rationally design the next generation. Right now, we're doing separate studies in all of these different types of cancers, and we actually then wonder at the end of the cancer, what if we did a combination, as Cyril was saying, what if we did these combinations in these other sites? Well, we don't even know if we would be shutting down the particular pathway in the group that we're in, and what the upregulated pathways would be, and what the differences would be among the different genotypic expression. I noticed in VEmurafenib, which I'm going to show you the data, the VEmurafenib study in colon cancer was a failure, but if you looked at serials data, there was only one cell line which had some modest activity of BRAF inhibition in it. But see, we're tantalizingly close, and Ryan showed you this data earlier this morning. So Ryan is leading a study for us of a combination of a RAF and a MEK inhibitor, both from GSK, and we've had some responses. Not as many as we would want, but we've had a few, and the few look incredibly real. Well, who are these people? So we've decided to expand the cohort and biopsy everybody on the clinical trial to figure out which of the pathways are being upregulated and downregulated. Of course, the resistance may be in the EGF pathway, as Ryan showed you, and not necessarily in the not getting around the RAF pathway by activating MEK. So the issue for us is we need to do more and more of these biopsy studies because we won't come up with these kinds of insights unless we do, and this has been confirmed as well. So in terms of future directions, we absolutely positively need to prospectively genotype the patients, but we also need to keep in mind that that's the way we talk about the patients. We actually need to figure out the way the biologists talk about the patients and group them accordingly as well. Targeting therapy after progression or stopping first-line therapy is key. I think if you get these patients too late in the course, it can be very difficult to administer any kind of drug, and the side effect profile, even if it's minimal, becomes overwhelming at that particular point for the patient and their family. Rebiopsying patients on phase 1 studies is critical, and we need to develop the protein biomarker capacity to do that and to do it often, as our lung group has been able to do at the general. And finally, this is critical because the cooperative group mechanism doesn't seem to be able to get off the dime on this. We need to develop a consortium of like-minded academic cancer centers that will prospectively do all of this up front. Because once the patient comes into us, if we're relying on all the patients that come into us from the community, it's going to be too late to do it at that particular time. We need to know at least what their genotypic status is going into the game from the moment they hit the door. And I'll stop there and take questions. Joe?
A while ago, there were studies of breast cancer where they evaluated new drugs before any other therapy, kind of a window opportunity. And it looked like there was no difference in survival when they did that. Is that something you're considering? Or?
Well, so stage 3 is very curable, and it depends on if we can't even tell reliably based on CT scanning whether somebody has stage 3 or stage 2. Stage 2 colon cancer, typically your average stage 2 colon cancer doesn't get any chemotherapy. Stage 3 does. We can't tell the difference between stage 2 and stage 3. So giving people upfront chemotherapy, like neoadjuvant chemotherapy for breast cancer, we'd love to be able to do that if we could reliably tell stage 2 from stage 3. So one of our active protocols here is, unfortunately, PET-CT, it gets so hot, the primary tumor is so hot that it overwhelms the ability to tell the nodal disease. And the lymph node size doesn't matter because you get these little micro perforations, and so you get 2 to 3 centimeter lymph nodes occasionally that are pericolonic that are negative. And so you might be giving chemotherapy to somebody who's got an 80 to 90% chance of being cured with all the attendant risks of doing that. So we need a better way to do it. What we're hoping, we actually have a protocol that's open right now looking at colon cancer patients with a PET MRI. With the PET MRI, you might be able to figure that out. And then in that particular setting for stage 3, you could potentially write a clinical trial where you're giving somebody a month of therapy before the operation. We're absolutely doing that for people who have liver metastases. Unfortunately, what's happening, though, is that because people who will present with liver metastases, because they have a shot of being cured, and because we see responses like I showed you early on, everybody feels really uncomfortable giving a targeted agent by itself when you could expect a response in about 40 to 50% of the patients, a 50% decrease or so in 40 to 50% of the patients that you treat. And that may influence the ability of the surgeon to get out all the tumor. And so you feel a little bit uncomfortable doing the neoadjuvant approach. But if we can do it in the stage 3 setting, particularly for folks who have N2 disease, who we know have a very high risk of relapse, I think everybody would feel more comfortable in that setting.
Bruce.
David, you mentioned the problem of dealing with anti-angiogenic therapy throughout the course of a patient's life. They've really dealt with this problem in myeloma and prostate cancer by continuing steroids with everything else. So could you just do your phase 2 studies with Avastin? You just accept the fact that people want continuous anti-angiogenic therapy.
You could. It's that there's two issues with that. One is the overlapping toxicities, which are always worrisome. Avastin isn't free of toxicities, or any of the VEGF inhibitors aren't free of toxicities. But I think people are going to move more and more to doing studies like that, particularly right after first-line chemotherapy, where patients are randomized to either continue bevacizumab plus a drug or placebo to get a sense of whether it's effective. We saw that with what was called the DREAM trial, where they did a bevacizumab plus or minus erlotinib. Unfortunately, erlotinib isn't all that effective as a single agent. But you could imagine, for instance, in the BRAF mutant patients doing bevacizumab erlotinib plus or minus VEmurafenib, or bevacizumab VEmurafenib plus or minus erlotinib, to kind of get at that question of is EGF the pathway of resistance for most of these patients.
Dave, is there any rationale for combining Avastin with aflibercept?
So total VEGF blockade of sorts.
I mean, if VEGF-directed therapy works, can you make it better?
There are probably better VEGF therapies. I guess the issue from my standpoint is it works differently in different tumor types. So renal cell carcinoma, clearly there seems to be a cytotoxic effect. In colon cancer, it seems to be more environmental and less cytotoxic, and even less cytostatic. It just seems to affect tumor growth in some way. And whether we can find better compounds or more complete blockade of that effect, I mean, it's possible, but I think we've pretty much exhausted. Bevacizumab has become our, so the fellows who are starting now feel about bevacizumab the way I did about 5-FU. So.
Maybe we'll end on that note.
Thank you.